Okay, the latest class speaks basically about humoral immunity, um, and the components of the humoral immunity are basically antibodies, complements, and cytokines. Okay, and we're also looking at methods that help us detect the presence or the functions of those three things. Okay, first of all, mm, what produces antibodies? As we know, B cells or memory cells when we have a secondary response. Leukocytes produce cytokines to help activate other cells, and liver produces the complement. Okay, antibodies are used in so many things around the body, such as in opsonizing foreign bodies that help, let's say, in phagocytosis, even sometimes create pores in the cellular structure of a bacteria. Cytokines are produced by, let's say, cells such as T helper cells that help activate other cells to cause a more amplified immune effect. And finally, liver produces the complement, and the complement system, one of its functions is that it acts as, or the things that exist within a complement system, such as 3,5A, would act as a tag molecule that attaches to the bacteria and tells the white blood cell or a phagocytic cell to attack this foreign body. Okay, that's just the way it is. And as we know, adaptive immunity, okay, is basically involving the cytokines and the antibodies, because as we know that B cells are part of the adaptive immunity, and T cells, which produce the cytokines, are part of the adaptive immunity. Passive immunity includes complement, as complements are always produced, irrespective of what stage we're at. Okay, so we're going to assess those three things. But before that, let's talk about how we can activate um, cells of the adaptive immunity so that they are able to produce antibodies, let's say. That's the first part that we talked about related to humoral immunity. And so B cells can get activated through two ways. Either they are activated by the help of the thymus, or they are activated by the help of themselves. And so how they do this, how this is done. So the first part is here, as it says, B cell activation by the thymus dependent antigen. And in this case, what we're looking at is a protein antigen. Okay. This process of B cell activation includes two signals. The first activation signal is when the BCR, or the B cell receptor, binds to a protein antigen, which is basically from the structure of the bacteria. Okay, that's the first way, or the first signal needed in the thymus-dependent activation of a BCR. As soon as this protein antigen binds to a BCR, it is phagocytosed inside and used, or broken down into smaller fragments, and then presented on its own MHC2 molecule, which is used in its further activation by the help of the T cell that we're going to talk about right now. Okay, so now that we have this protein antigen bound to MHC2, okay, which we have originally got from the binding of BCR to protein antigen, which is brought inside the cell and then presented on the MHC2, is now used to be part of the second activation signal of the B cell that is activated inside the thymus. Okay, طيب هسه inside the thymus, okay, what's used to activate B cells? T helper cells, okay? T helper cells have this antigen on their surface called CD4, okay? And so this is just a labeling of what type of cell it is. So just for your information, the cells that would activate a B cell containing an MHC2 is a cell that contains a CD4. Okay, and so how do we remember these specific combinations of of receptors, or let's say specific handshakes, we always remember that 4 times 2 equals 8. And so to know exactly what binds to a cell that has an MHC2, we need a number such as 4 to be multiplied by 2 to give us an answer of 8. So in case we want to activate a cell that has an MHC2, okay, the cell that would bind is a CD8, okay? But in this case, sorry, I meant a cell that would be killed, okay? It'll be a CD8 because CD8s do not activate, but they kill, okay? But when they bind and shake hands with a cell that needs to be killed, it needs to bind to an MHC1. So 8 times 1 is 8, okay? Now let's go back to activation. CD helper, CD4 T helper cell, would cause the second activation signals by shaking hands with the B cell by 3 ways, okay? The first way is, as we know, TCR binds to MHC2. The second way, CD4 binding to the same MHC2 on a different place, okay? So that's 2. The third one is that a CD40 ligand found on the CD4 binds to a CD40 receptor on the B cell. Okay? طيب هلا when this happens, cascades inside the the B cell gets activated. Now the B cell is able to produce IgG antibody, which is the long-lasting immune response. Why it's long-lasting is because IgGs have a long half-life within the body, so they are not broken down easily, okay? The second thing that we are going to discuss is the activation of the B cell by thymus-independent process. And what we mean by here is that the B cell is getting activated without the help of the thymus. And in this case, it differs in 3 ways. First one, we need a sugar antigen to bind to the BCR. And this sugar antigen binds to the BCR, causes the first activation signal. Okay? By the way, you're seeing. Okay? First activation signal. And then the second activation signal is what? What? 1 second. Okay, wait. Because you're here. Okay, so the first signal, sugar antigen binds to the BCR. The second activation signal is that there's a receptor on the B cell called the toll-like receptor, binds to that same sugar antigen, maybe just a different molecule from the blood, and so causes the activation of the B cell. And what is released here is the IgM. The IgM is an antibody that causes immunity to be shorter than the immunity of IgG, okay? And so now we know to differentiate between thymus-dependent and thymus-independent. Thymus-dependent would need a protein antigen. It needs the protein antigen to bind to the BCR, but then we have T cells that are present instead of having it to be activated on its own. And then the production of IgMs here, and on the top you need an IgG, okay? So these differences you have to know between thymus-dependent and thymus-independent, okay? Protein antigen, T cell present, IgG. Sugar antigen, T cell not present, IgM. And then, of course, the different receptor interactions. This is just a summary of what we just said. This is just a scheme to tell us the life cycle of a B cell, and then it reaching to the point where it produces different antibodies. And then now we are trying to actually test using methods or tools in the lab to actually tell us whether the immunity of a B cell is working or not. And why do we want to check is because sometimes we are very suspicious over the health of an immune system, and this is a suspicion that arises from the increase of bacterial infection when somebody gets sick so many times. And so we are going to use what we have originally studied to actually test and look into the health of an immune system. What we do usually is stimulate the B cells normally by either antigens or mitogens. And if we remember, antigens are specific things that activate specific things, and mitogens activate generalized, non-specific things, okay? And so let's say we use either antigen or mitogen. A B cell gets activated. Now, when a B cell gets activated, it proliferates. It has BCR expressed right now, and then it produces antibodies. And for every single stage of the life cycle of a B cell, we have a way to detect whether it's happening or not. And so the division of a B cell to produce many B cells, we would use CSFE or Burdu. As you remember, CSFE decreases as the generations of proliferation increases. Burdu is just a way in which we can detect the presence of so much DNA as the cells proliferate and increase the number of DNA within the B cell community. The second thing is that when we have a BCR expression after proliferation, we can detect the presence of a BCR by cytometry, flow cytometry. We basically label the BCR with an antibody that is attached to a fluorescent molecule and expose it to flow cytometry to detect the presence of BCR. And then finally, we can detect the products of this activation where B cells produce antibodies. And so to detect the presence of antibodies, we use ELISA. And ELISA uses a sandwich system of antibodies that either are attached where the secondary antibody of this sandwich is either attached to a fluorescent molecule or an enzyme that can digest a substrate that we add later to change color. And so once we detect those changes of either light or color, we can detect the presence of the antibodies, okay? Now, how can we activate a T cell? Okay, so why are we also now talking about T cell activation and B cell activation? To remind you that these cells are the ones that produce the components of the humoral immunity. B cell activation is used to produce the antibodies. T cell activation is used to produce the cytokines that we are actually assessing in this presentation that started here. We talked about antibodies. Now we're going to talk about the cells activation, T cells activation that produce the cytokines. Now, T cells needs 3 signals to get activated. And as we remember that an APC, an antigen-presenting cell, in this case a dendritic cell, activates a naive T cell into either a cytotoxic T cell or a T helper cell of different kinds. And I'll tell you how these specifically get produced, okay? So the first signal that happens between T cell, the naive T cell, and antigen-presenting cell is when they shake hands using TCR from the T cell and an MHC2. Okay, so if an antigen-presenting cell uses MHC2 to shake hands with the TCR of the T cell, this means that the cell is going to be a T helper cell. Why? Is because 4 times 2 is 8. And so if we know MHC2 is going to be here, then we know on the opposite side, a CD4 cell will be produced, okay? That's the first signal. TCR combines with MHC2. Okay, the second signal is a co-stimulatory signal where CD28 from the T cell combines with CD80 or CD86 on the antigen-presenting cell or the dendritic cell, okay? That's the second signal. The third signal is the release of cytokines, okay? And so antigen-presenting cells release some sort of unique cytokines to tell exactly what this naive cell is going to be, okay? So if, let's say, an IL-2 is released, I think a T helper 1 is going to be produced, okay? That's the third signal. So first signal, TCR emits C2. Second signal, CD28 combines with CD80 or 86. Third signal, cytokines release. And so when antigen-presenting cell releases cytokine, a receptor on the T cell detects the cytokine, and according to what type of cytokine is detected, this T cell becomes that, okay? This is easy. We took it in microbiology, but I'll just go over it quickly. Usually antigens that activate T cells allow them to produce cytokines in an acceptable amount, which helps us to actually fight the bacteria. Sometimes cytokines are overly produced and cause a shock more than a normal response. And these are super antigens. And if you remember TSST-1 from the Staphylococcus aureus, causes a great response that leads to a shock, a septic shock, basically. Okay, so another toxin or a super antigen is the staphylococcal enterotoxin A. Okay, so just memorize those two examples as super antigens. You may be asked. How does it detect, just like we detected the activation of B cell using CSFSE, Burdu, what was it? Flow cytometry and ELISA, we can use the methods that we have learned to detect the activation of T lymphocyte as well. And for example, what can I say? The methods that can be used in this case, let's say for cytotoxicity, we can use the ROS tests, such as far as I can remember, MTT, for example. Yes, I think so. MTT was used to detect cytotoxicity. Or you can use the chromium, I think, the chromium radiation method, just so we can detect the activity of this T cell. All right, moving on to the complement activation. The complement system is basically a system that, as I said, helps to tag cells that are foreign to our system, and also can help us in actually causing pores within the structure of the bacteria, such as the use of the MAC complex, which is the membrane attacking complex. And this complex comes about through three different ways: the classical pathway, alternative pathway, and lectin pathway. And I wouldn't dive deep into this right now, but if you'd like, you can go ahead. But the whole reason of this complement activation is to produce the MAC complex. Now, complement activation and how we can detect if complement works or not. Now, just to keep track where we are and why we are talking about complement right now all of a sudden, because I have said in the beginning that the humoral immunity depends on three factors: antibodies, cytokines, and complement. And complement was produced by the liver in that first slide that we have discussed. And so now we are discussing complement. Complement, yes, produces MAC complex, which is necessary to attack and help us fight the bacteria. Now we can detect if it's functional or not. And so we may be suspicious over the function of the immune system because of so many bacterial infections. And so how can we do this is by two tests. The first one is a complement activation assessment. Either it can be the 50% hemolysis CH50 or the radial hemolysis, okay? And so what CH50 means is basically we test the amount of patient serum or patient antibodies needed to lyse 50% of the erythrocytes used to prove the function of the complement map. Okay, so that's the first way. And the second way is the radial hemolysis. In this case, we use sheep erythrocytes that are fixed. Okay, I think cells here are not fixed, but cells here are fixed. Now, the second method that we can use is just the detection of the presence of the complement proteins. And this is going back to the methods that we have discussed related to detection, such as radial detection, ELISA, etc. Okay. Now we can talk about antibody diagnostics. These are just the structure of the antibodies. Just know that these are the IgG antibodies that are generally found free in the blood, and they cause long immunization. And these are produced, as we remember, from the thymus-dependent activation of B cells. IgMs, on the other hand, are produced from the thymus-independent activation of B cells, and they cause a shorter immune response. And IgMs are basically the BCRs that are found on B cells and released when the B cells are activated. I think these are just facts. You can just go ahead and look at it. What is valency? What is affinity? What's avidity? Valency, how many are bound to antigens at the moment. It's the measure of how many antibody-antigen complexes are present at the moment. Affinity, the strength of one antibody bound to one antigen. Avidity, the sum of those strengths. Okay, again, valency, how many are bound at this moment. Okay, affinity, the strength of binding between one antigen and one antibody. Avidity is the sum of all the strengths. Okay. Now, sorry. Antibodies. Okay. How can we detect their presence? And so, when are we suspicious whether antibodies are present or not? During infectious diseases, during immune deficiencies, during proliferative disorders, during autoimmune diseases. And so all of those cases, we are curious if the antibodies are present or not. And so different cases have different tests that we can use to detect the presence of antibodies. First one, infectious diseases, we can either use ELISA or a Western blot, and those two can give us whether the antibody is present during an infectious disease. Okay. Now, immune deficiencies. During an immune deficiency, we can use also two methods. The first one is basically immunophilometry. This is where we use light to scatter off complexes of antibody-antigen. So we expose the sample that may be or may contain an antibody-antigen complex and shine light. And so we detect this light that scatters off those. And so the more light that scatters, the more of those complexes, and the more we are happy that antibodies are present. Second ways, or many ways, are those specific ways that we can use to detect those antibody antigen complexes. And then in proliferative disorders, we can use electrophoresis of serum proteins, okay, or immunofixations, okay. I'm not going to dive deep into those, but just read it quickly. And autoimmune diseases, we can detect antibodies using class 8 methods, and these are going to be discussed in the future. So how can we produce monoclonal antibodies? We are going to be taking cells from the mice that we use for our experiment, we will take B cells that are able to produce antibodies, and we will take cells that are cancerous and fuse those two. Why? Is because cancerous cells cannot produce antibodies. Yes? And B cells, cancerous cells cannot produce antibodies and cannot activate themselves. And B cells can produce antibodies, but they cannot produce it in large amounts. So why are we combining those two? Is because we need cells that produce antibodies in a large amount, hence cancerous cells, and we need cells that can activate themselves on their own, hence B cells. So when we combine those, we have those two factors in one cell. And so we use a hat medium that will only allow the growth of this hybrid cell that we actually need. And then we take those hybrid cells, clone them, have a lot of them, produce a lot of antibodies, monoclonal antibodies, and then we can use it to actually inject in somebody who needs them. Okay, this is just a summary of this slide. Okay, so how can we use antigens as antibodies as antigen finders? Okay, and so we have studied this already. This is just basically telling us where can antibodies be used to find antigens in what methods. And we have already discussed this. We can use antibodies that are bound to fluorochromes, okay? And these antibodies can be specific to a specific antigen, okay? And so once it bounds and we explore it to light, it shines. So it tells us that there is an antigen present at that spot. Or we can use enzymes. We can attach an enzyme to an antibody, and this antibody can bind to that specific antigen that we want. And so we add a substrate, that substrate gets digested, the color changes, and then we can detect the presence of that antigen. Same here as a radioactive isotope or chemiluminescence, yes? And so there's also methods that are direct or indirect. And she said and asked in the class, what is direct and what is indirect? Indirect is basically using a secondary antibody, and a direct, we don't use a secondary antibody. What am I referring to? When you look at ELISA in the previous class, you can see that there's a sandwich of antibodies that are bound to that specific antigen, yes? So you will have, for example, a capture antibody, the antigen, the detection antibody, which is the primary one, the secondary antibody, which is bound to those bodies that we discussed here, the fluorochromes, the enzymes, etc. Yes? So now direct basically has the capture antibody, the antigen, and then it has just one antibody that is bound to that antigen and bound to that fluorochrome or enzyme. The indirect is using just yet an additional antibody. Capture, antigen, detection, and then the secondary, okay? This method, basically, what she said here is like enzymatic method, system envision is basically a structure of antibody formation that is used to detect things that are very hard to detect within the body. That's the only thing I remember. Okay, here it basically says, when can we use antibodies as a form of therapy? In case of humoral immunity deficiencies, okay, basically we can't produce antibodies, we just infuse antibody, okay? In case of toxin neutralization in emergency, like there's a toxin in the body and we need antibodies to immunize it, but or maybe antibodies are not sufficient enough, we administer antibodies intravenously so that it neutralizes that toxin. And in case of autoimmune diseases, we also infuse or administer antibodies, okay? So that they can compete with the antibodies already present. Okay, here basically we are just saying that we are changing the structure of an antibody to actually fit a certain function. Okay. Okay, here it just says, what can an antibody be used in to attack, let's say, a tumor cell or a cell that is foreign? For example, here we need an antibody to bind that to that tumor cell. And so once it's bound to that tumor cell, that killer cell is now able to bind to that tumor cell and then cause a death effect. Or in case, let's say we need a lysosome to bind to this tumor cell to release all of its enzymatory substances. And so the only way these two would bind is using an antibody. Okay, so just this last slide, I had to just do some research on it. Okay, so in normal ways, in normal cells, uh, the antibody, or no, sorry, the protein, HER2 found here. Okay, what is this? This is a breast cell, okay? Normal breast cancer cell, okay? So it's just a cell that is usually present within the breast tissue, okay? Normally, HER2, what it does is stimulates the proliferation of the breast cell, okay? In abnormal, um, highly cancerous cells, HER2 is overly expressed. And so if it's overly expressed, it overstimulates those cells to proliferate and hence cause cancer. Okay, so how can we detect this? There is a test called Hercept test, and it uses Herceptin antibodies. Okay, Herceptin antibodies would help us detect the overexpression of HER2 receptors that cause an over proliferation signal for the breast cells, causing them to be cancerous. And the Herceptin is the antibody that we use binds to that HER2. And so once we detect the overexpression of HER2, now we know that this is a cancerous cell. Okay, that's it. Okay, good luck in your orals, and maybe see you guys in the next one.